All right, welcome back, everyone. So we just kind of left off talking about memory as types and stages. Let's talk a little bit about some of the other processes that are involved. So psychological research has produced a lot of information that looks at the type of processing that's involved in how we remember information. And so in order to successfully um, get information out of long-term memory, there are three important things that need to happen, three important processes, and those are the processes of encoding, storage, and retrieval. Let's talk about them for a minute. Encoding is the process by which we place the things that we experience into memory. So if we don't encode the information, we won't ever be able to remember it. So, for example, this is like when you hear someone's name for the first time and you don't do anything with that information. Well, this is what happens when we don't encode that information in a meaningful way to be able to retrieve it from long-term memory later on. Now, many things that we experience we don't end up encoding. So we tend to focus on encoding the things that we need to remember. So one of the challenges is that sometimes, though, there are things that we need to remember that perhaps don't come naturally to us. And so how do we do that? Well, there's something we can do called elaborative coding. And so that's the process that when we process new information in ways that make it more relevant or meaningful. We try and relate it to our information in our own life or connect new learning to old learning. This helps us to be able to remember, to encode some information. Now there was a guy, and his name was Ebbinghaus, and Ebbinghaus found that he did these studies of memorizing lists of words and then looking at how easily it is for people to forget this information. What he ended up finding was that there's a very predictable way that information decays in our brain, and so he came up with this idea of what we call the forgetting curve. So what we find, if you can look at the picture at the bottom, is that memory decays rapidly at first, and then after after 1 or 2 days, like that's where we lose most of the information is when the first couple of days. After that point in time, the retention kind of levels off. So by about day 5 or so, we're kind of just we're on the path to leveling off. So by about day, well, what does it look like, about day 8 or so, that we are basically just, we don't forget much more. After that, it pretty much just levels off. So most of the information we forget it right off the hop. So what this curve suggests is you should try and review the material that you've already studied right before you take the exam. So that way you'll be more likely to remember the material during the exam. Now, Ebbinghaus also found another important thing, and we've talked about this at the beginning of the term, but this idea of spacing out our learning. So the fact is that our learning is better when the same amount of study is spread out over periods of time rather than when it occurs very closely or all at the same time. So practically what this means for you is, instead of cramming at the very last minute, if you study continually throughout the semester, you will actually remember more at the time when you need to take the final. Spacing really is your friend. Ebbinghaus also played around with the idea of overlearning. And overlearning is when we continue to practice and study even when we think that we've mastered the material. Now, there's a couple reasons why we want to do this. One, we frequently think that we've already mastered the material, but when we get to the exam, we may realize, like, oh crap, I really didn't. So, overlearning is that we keep studying and reviewing even if we think we already know all the information. This increases the chances that we've solidly encoded the information and that we're storing that information into long-term memory. Now, if we've encoded the information, well, that's great. It's no good to us if we've encoded it, but we can't get it out of our mind. And so retrieval is the process of reactivating this information that's been stored in memory, going into that filing cabinet and pulling out the file. One of the easiest ways for us that we kind of see that we've had a bit of an issue with retrieval is what we call the tip of the tongue phenomenon. So the tip of the tongue phenomenon is when we're certain we know something we're trying to recall, but we can't quite come up with it. And that's when we feel like we've got the right answer just at the tip of our tongue, but we aren't able to bring ourselves to fully kind of say that. So the information is stored in memory, we've encoded it, we're just having a heck of a time retrieving it. Now, what we do know about retrieval is that we're more likely and more able to retrieve items from memory when the conditions at retrieval are similar to the conditions under which we encoded them. So, what does that mean for you? Well, we came up with this idea, or psychologists came up with this idea of context dependent learning. And so, what we found is that there's an increase in retrieval when the external situation where we learn the information matches the situation in which it is remembered. So, for example, if you study for an exam in a situation that's similar to the actual exam you're going to take, you'll find that your learning is much better and you'll do much better. So, you know, a library might be a good example, or in our case, where we usually just learn the information in one spot and then have the opportunity to retrieve it, that is actually most helpful because then you are learning the information in the context in which you're going to have to be retrieving it in the future. We've also found, though, that there is a state-dependent component to it. So when we learn information in, I guess I should say this, our retrieval is superior when our physiological state or psychological state is the same during encoding as it is during retrieval. So what does that mean simply? Well, if you are happy and in a good mood when you come to your lectures and you're learning that information, and then on the exam you're in a good mood and you're happy when you've got to retrieve that information, well, if there's a match between your state and in learning and in retrieving, well, heck, you're going to do much better, and your recall will actually be significantly better than otherwise. It's also easier for us to recall unpleasant memories than pleasant ones when we're sad, and it's easier for us to recall pleasant memories than unpleasant ones when we're happy. So this kind of is a little bit of evidence for that, but also, in actual state-dependent learning tasks, the state, excuse me, in which you learn the oh my goodness, the state in which you learn the information is very important for retrieval. Ebbinghaus, in all of his kind of work, also noted what we call a serial position curve. And really what this is saying is that Ebbinghaus, in all of his studies, he found that people were really super good at retrieving information at the beginning of a word list and then at the end of a word list. So Ebbinghaus would make people memorize random word lists, all right? So some of them were nonsense words, some of them were just random words that were thrown together with no meaningful connections. And what he found is that people were always better at retrieving stuff at the beginning of the list and at the end of the list, but everything in the middle, that was really kind of poor retrieval. And so what he came up with was that this is due to two effects. The first one is what's called the primacy effect, and that's the tendency to better remember things that are presented early in a list. And the second thing is the recency effect, and that's the tendency to better remember stimuli that are presented later in a list. So we remember things at the beginning and at the end of the list much better than the stuff in the middle. And here's how it kind of looks. So the probability of recall is significantly higher for things that's in the beginning of the list, and so this, is compared to things in the middle, and things at the end of the list also kind of improve. Now, why do you think this is? So if we think about short-term memory and some of the processes that are involved in keeping information in memory, there's got to be a clue here for us, right? Well, there is. Because we can keep the last words that we learned presented in short-term memory by rehearsing them before the memory test begins, they're relatively easy to remember. Also, when we begin to hear words right at the beginning of the list, we start to rehearse them. So it makes it more likely that it'll be moved from short-term memory to long-term memory. So in other words, our rehearsal at the beginning, as well as the fact that in short-term memory, usually at the end of the list, we're within that 18 seconds, so we're able to kind of keep that stuff alive. For both of those factors, that's why we have both a primacy as well as a recency effect. Now, for words in the middle of the list, well, good luck, because if you're still busy learning the stuff at the beginning of the list by the time you get to the middle, so that's likely why they are much less likely to be moved into long-term memory. In the same breath, sometimes our existing memories can actually influence our new learning and make it challenging. And this impairment of our learning can happen either in a backwards fashion or a forwards fashion. So if it's happening in a backwards fashion, it's retroactive. So retro, thinking of like the good old days, right? And retroactive interference happens when learning something new impairs our ability to retrieve information that we learned earlier. So for example, you get a new credit card and you get a new PIN number. when you get that new PIN number, sometimes you just automatically forget the old one that you've learned. So if you ever called, asked to recall the old password or the old PIN, it's a very challenging thing. Well, in this case, when you learn something new and it interferes with things you've learned before, that's retroactive interference. In the same breath, it can also happen in the opposite direction. And this is what I tend to find most is proactive interference. And that's when earlier learning impairs our ability to encode information that we may try to learn later. Now, in my line of work, every month we basically have to change all of our computer passwords. And so I find that I struggle with proactive interference because I've learned that old password so well that when I'm trying to learn a new complex password, it takes a lot more mental effort. So that is called proactive interference. Here's a little bit of a chart for you just about some of the important kind of pieces that and the important ideas and techniques that you should be aware of that we've just covered in this slide. So here's a nice little chart version of all of the previous slides. It gives you a description and an example. I kind of like them. Now, it's important for us to chat a little bit about the structure of long-term memory. So memories that are stored in long-term memory are not isolated, but rather they're linked into categories. When we're talking about categories, we're talking about networks of associated memories that have features that are in common with each other's. So forming categories and using categories to guide behavior is a fundamental part of our human nature. Our associated concepts within a category are connected through spreading activation. So what that is that activation of one element of a category activates other associated elements. So to show you this, here you could start with something like dog, right? And spreading activation would be that we would say like, well, a dog can be a pet, a dog can also bark, a dog can is opposite from a cat, a dog is an animal, a dog is a friend, a dog is sweet, right? And then from each one of these, kind of large, bigger concepts, we then can actually have other connected categories. So this is what's called spreading activation. So if you just say dog, well, then we go pet. Well, if it's a pet, then there, other pets are bunnies or hamsters or gerbils or cats, right? If you think bark, you might think growl, right? It's like aggressive dog behavior. You might also associate dog with cat, and so then you might think kitten and black, or dog with animal, and so then you might think, well, here's some other animals. So this is a diagram of what spreading activation looks like in long-term memory. So it's not just like a neat little file that's in one kind of place. Our memories are connected into these networks. Now, some categories have a defining feature that must be true of all members of that category. But most categories that we have are not so well defined. So memories or members of the category may share some common features, but it's really impossible to define which are or aren't really members of the category. And so when we look at categories, we look at what is the most prototypical member of a category. And so a prototype is the member of the category that's most average or typical of that category. So for example, if we think of pop, right, the most prototypical pop that we tend to think of first is, like Coke or Pepsi. Maybe we'll think like Sprite or 7-Up, right? Those are things that are most, prototypical. But maybe now we'll also think I don't know, like Canada Dry, right? Like that's not maybe as as prototypical, but it's connected to that category. So when we're thinking pop, we think like, what's the first things that comes to mind? Those are generally the things that are the prototypes. So for example, when we're retrieving information, we're going to retrieve information that is prototypical of a category much faster than when we retrieve information that's less prototypical. For example, if you're asked to think of a dog, you're likely going to think of like a golden retriever or a lab or a poodle much faster than what you're going to think of a dingo, right? Even though a dingo may actually fit within this category of dog. So prototypes are very, very common, and we rely on them quite a lot. In addition to prototypes, we also have schemas. And schemas are patterns of knowledge that help us to organize information. We tend to have schemas about objects, about people, about events, about social groups. So for example, if I asked you, what is a schema for going out, to a restaurant for a meal? Well, you would say to me things like, okay, well, basically, if you go out for a meal, the same things happen, right? You go into the restaurant, you wait to be seated. Once you sit down, they hand you a menu. They go away for a couple minutes, and then they come back and they ask you what you want for a drink, and then they go away for a minute, and then they'll bring you your drinks, and then they're going to ask you what you want to order, and then they go away for a while, and then they bring you your food, right? There's this schema of how it works when you go to a restaurant, and when our schemas don't fit, right, or our experience doesn't fit our schema, we generally think it's a bad experience. Right? So schemas are very important because they help us to remember new information, and they provide an organizational structure for us for how these things work. There's some examples for you there too of different types of schemas. Now, not only is information stored in the brain in a vague sense, information in long-term memory is stored in specific areas in the brain. Now, the ability to maintain information in long-term memory involves a gradual strengthening of the connections among the neurons in the brain. So when pathways in these neural networks are frequently and repeatedly fired, the synapses become more efficient in communicating with each other, and these changes create memory. And we call this change, we call it long-term potentiation. So that's when these synaptic connections between neurons are strengthened because of frequent stimulation. Now, we know that there are drugs that block, that can block that long-term potentiation. And what we found is that when LTP is blocked, it reduces learning, whereas drugs that enhance that long-term potentiation actually increase learning. Now, because the new patterns of activation in the synapses take time to develop, long-term potentiation happens gradually. It's not like you learn something and all of a sudden, boom, those neural connections are firing at full tilt, right? So the period of time in which long-term potentiation occurs and in which memories are stored, this is known as the period of consolidation. All right? And consolidation occurs as a result of changes in the synapses, which suggests that chemicals, in particular neurotransmitters and hormones, must be involved in the memory process. So we know, for example, that glutamate is perhaps the most important neurotransmitter in memory. For example, we know when animals, including people, are under stress, more glutamate is secreted, and this glutamate can actually help them to remember things. We also know that serotonin is secreted when animals learn, and epinephrine may also increase memory, particularly for stressful events. Estrogen, as well, may also be involved in memory formation because women who are experiencing menopause, which results in a reduction of estrogen, frequently report memory issues. So we have a fairly good idea that glutamate, serotonin, epinephrine, and estrogen play an incredibly important part in the memory consolidation process. Now, memory is not confined to the cortex, which is actually a pretty surprising type of thing. But it occurs through sophisticated interactions between new and old brain structures. Now, one of the most important regions in the brain for explicit memory is the hippocampus. And so the hippocampus is located right here. It's part of the limbic system. And it serves as a preprocessor and an elaborator of information. The hippocampus helps us encode information about spatial relationships, the context in which events were experienced, and the associations among memories. It also acts as a relay station that holds memory for a short time and then directs the information to other parts of the brain that actually do the rehearsing, elaboration, and long-term storage. While the hippocampus is handling explicit memory, other parts of the brain handle other memories. So the cerebellum, the old brain, the oldest, structure in the brain, is responsible for those implicit memories, the procedural things that we know how to do. We know that the amygdala as well is particularly involved in emotional aspects of memory, and in particular those types of memories that involve fear. Now, evidence for the role of different brain structures in different types of memories come in part from case studies of patients who suffer from amnesia. So when we talk about amnesia, it's a memory disorder that involves the inability to remember information. Now, just like interference with learning things, amnesia can work in either a forward or a backward fashion, which would affect retrieval or encoding. So problems remembering things that have happened in the past is called retrograde amnesia. All right, so we're having difficulty remembering things that have happened before. And what happens then is there's a certain point in time that a person is unable to retrieve events that have occurred before that time. Now, retrograde amnesia is usually most severe for memories that occurred just before the trauma than it is for even older memories. And this type of amnesia is due to trauma to areas that are involved in long-term potentiation and consolidation processes. Okay, so LTP and consolidation are connected here to retrograde amnesia. On the other hand, we can also have anterograde amnesia. So that's amnesia going forward from a certain point in time. And what happens with anterograde amnesia is that there's an inability to transfer information from short-term into long-term memory. So organisms with damage to the hippocampus develop a type of amnesia that works in a forward direction, and this affects encoding. So people with anterograde amnesia have problems forming new memories. Now, while anterograde amnesia may stop you from forming new conscious memories or explicit memories, it may be possible to form implicit memories, such as procedural memories like riding a bike or learning how to play chess or doing something like that. So you may learn implicit things, but explicit memories is really where the challenge lies. Now, this also goes to show you, just like the previous slides have said, that there's no one area in the brain that is responsible for all things memory related. And that's probably a good thing because injury to the brain does happen. And so if memories were only consolidated in one spot, boy, wouldn't that just be bad if that part of your brain was ever injured. Now, this was partly, due to Carl Lashley's work. And in the early 1920s, Lashley would systematically remove brain tissues from rats to try and find where memory was stored in their brains. And so what he found is that no matter how much of the brain he ended up removing, rats would maintain at least some memories. And he judged that based on their memories of being able to complete a maze. So with rats, oftentimes in scientific studies, you would teach a rat how to complete a maze. And so they would learn that at the end of the maze, there's a treat or some cheese or something. And so they would learn how to do these mazes quite quickly because they would like to get the treat. And so what ended up happening was, so he would teach them the maze, he would cut parts of their brains away, and then he would see if they could still remember it. And he would find that they were still able to remember pieces of it, no matter how much of the brain he cut away. So this led him to conclude that memory isn't located in a single place in the brain, but rather is distributed around it. And that's also what case studies tend to show, is that memory is not located only in one aspect of the brain. It's located everywhere. Now, obviously with some of the case studies, like the case of HM, who had severe anterograde amnesia, we've realized, well, heck, like the hippocampus is pretty important for explicit memories. But at the same time, we also learned that HM was able to gain new procedural knowledge, so new implicit memories. So again, animal studies as well as case studies confirm that memory is not just located in a single place in the brain, but is rather distributed throughout the brain. So obviously, if there's these pieces of long-term potentiation, consolidation, retrieval, and all these things, are there ways that as people we can enhance this for ourselves? This is obviously, the question we always like to ask. And so a lot of studies have looked at, are there ways that we can enhance memory? And the reality is we found that there's actually very little evidence for the effectiveness of a wide range of psychopharmacological options for improving memory. So what we found is that in general, memory supplements are usually no more effective than drinking a sugared soft drink, which also releases glucose and thus improves memory slightly. So if you have the option between taking some Ritalin or having a Coke, go for the Coke. It'll be, just basically it'll give you about the same effect. Now, it's not to say that one day we won't have these, drugs where we can do that, but right now, using drugs to improve memory has not actually been found to be effective. Okay, I'm going to pause here and we'll pick things up again in a minute.